My name is Jennifer Obike. I'm an undergraduate researcher in the Obike research group that primarily focuses on the use of natural products for pharmacological purposes. Today, I'll be presenting on the media and strain study for enhanced production of cis-enone resorcylic acid lactones. I'll be discussing fungal secondary metabolites and the challenges that is faced while working with them, resorcylic acid lactones, the method used, results, and the future works of this project. The use of natural products in terms of fungal secondary metabolites as a means of medicinal purposes increased after the discovery of penicillin. Fungal secondary metabolites are produced in fungi due to different environmental factors. These metabolites can be isolated and used for medicinal purposes. However, the challenges of working with natural products or secondary metabolites are low yield in nature, slow growth of fungal cultures, and unpredictable amount in isolation. That's why our research goal is primarily focused on the resupply of these compounds. I'm going to be focusing on resorcylic acid lactones, RALs. RALs are a class of fungal secondary metabolites that are known to have pharmacological properties. I'll be focusing on two types of cis-enone RALs, hypothemycin and 5Z7 oxozeaenol. These two structures are very similar. However, there is a structural change on the location on the red. Hypothemycin has an epoxide group, while oxozeaenol has an alkene group on that specific location. These cis RALs are known to have inhibitory effects on protein kinases such as TAK1 enzyme, which can have anti-cancer activity. This was a general method that was used to obtain the extract material of hypothemycin and oxozeaenol. First, we screened over 536 fungal cultures and found these three strains to have either of the both cis-enone RALs. And they were grown on three different medium, rice, oatmeal, and cheerios. To first extract the compounds, we remove the fats and sugars through separation by having a partition of chloroform and water, and for the second partition, a 1-to-1 ratio of acetonitrile and methanol with hexane. We further separated the extract via flash chromatography and observed it on UPLC mass spec to ensure that we have the presence of both cis-enone RALs. The cultures were observed under UPLC mass spec to determine the presence of both RALs compounds. For the fungal strain MSX7A495, all medium had both RALs compounds. For the fungal strain MSX63935, the rice and oatmeal medium only had oxozeaenol. However, for MSX45109, all of the medium had little to none of hypothemycin and oxozeaenol. This showcased the results for the relative abundance of which medium enhanced the production of RALs compound. For hypothemycin, the oatmeal medium on fungal strain MSX7A495 was the best enhancer. For oxozeaenol, the fungal strain MSX63935 was further enhanced by rice. With the results obtained from the relative abundance, we proceeded into purifying the compounds. For hypothemycin, we used all the medium for the fungal strain MSX7A495. However, for the large-scale experiment, we'll be focusing on the oatmeal medium. The cultures were further separated on ESCO to obtain the hypothemycin-containing fraction. We then reconstituted that in 2 cycles of 12 ml and 6 ml methanol, which was centrifuged. The precipitate was collected, and we obtained a 1,040 milligrams of a greater than 94% pure hypothemycin. The same process was used to purify oxozeaenol. We used MSX63935 grown on rice. However, we did an additional cycle of acetonitrile and obtained 760 milligrams of a greater than 94% pure oxozeaenol. To make the long story short, the fungal strain MSX7A495 grown in oatmeal enhanced the production of hypothemycin. The fungal strain MSX63935 grown on rice medium further enhanced the production of oxozeaenol. Fractionation with the use of flash chromatography along with resuspension and centrifugation technique, proved to be a better alternative for purifying large amounts of material. HPLC has been the technique that was previously used to purify compounds. However, this technique requires a large amount of solvent and is very time-consuming. Resuspension and centrifugation technique could be considered green chemistry, as we're minimizing the use of large amounts of solvents when it comes to purifying samples. This result that we obtain for this project will be used when scaling up the experiment. With the resupply of both compounds, this will enable a series of semi-synthetic reactions to be taken place. This in turn can enhance the bioactivity of the compounds. Thank you so much for your time and attention.